Assalamu alaikum. Today the topic is Carnitine shuttle system. This system represents a mechanism by which long chain fatty acids, which are impermeable to the mitochondrial membranes, are transported into the mitochondrial matrix for the purpose of beta oxidation and energy production. Carnitine is derived from diet or biosynthesized in our body. It can be biosynthesized in the mitochondrial matrix. It can be biosynthesized from lysine and methionine, whereas short and medium chain fatty acids can pass through mitochondrial membranes. This is a little introduction to the carnitine shuttle system. So here we can go through this system, that how long chain fatty acids can go through the mitochondrial matrix and how carnitine is biosynthesized. So here the long chain fatty acids come from this. So first we talk about the mitochondria. It has two membranes, the outer mitochondrial membrane and the inner mitochondrial membrane. Below these membranes the mitochondrial matrix is present, and between these two membranes the intermembranous space is present. So here the long chain fatty acids comes from the cytosol, and acyl CoA synthetase enzyme is present here. Okay, what this enzyme actually do? Acyl CoA synthetase enzyme actually do, it neutralizes a couple of cofactor and utilizes the ATP and CoH and make fatty acyl CoA. Okay, once it makes fatty acyl CoA, the CPT1 is present in the outer mitochondrial membrane. CPT means carnitine palmitoyl transferase 1 is present. Okay, what it does? What was the work of CPT1? CPT1 add the carnitine to fatty acyl CoA and CoH and makes fatty acyl carnitine. It gives one carnitine to the fatty acyl CoA. CoA, instead of CoA, it gives carnitine. So it transfers from the outer mitochondrial membrane to the intermembranous space. Here the CACT is present. CACT is carnitine acyl carnitine translocase is present, which transfers the fatty acyl carnitine into the mitochondrial matrix. Okay, in the mitochondrial matrix the CPT2 is present, which takes the CoH and add CoA to fatty acid chain and makes a loose carnitine. You people can see here the CPT2 enzyme is present in the mitochondrial matrix. It takes the CoH and produces fatty acyl CoA and makes a loose carnitine. This fatty acyl CoA will undergoes the beta oxidation, and as end product we get acetyl CoA. Okay, this carnitine, this loose carnitines will move to the cytosol through CACT enzyme and enters into the cytosol. Enters the cytosol for another long chain fatty acid and used by long chain fatty acids as a carrier molecule. Carnitine can be regenerated in another way by carnitine acetyl transferase enzyme, which is present here. This enzyme takes the acetyl CoA from the beta oxidation and moves acetyl CoA and moves the acetyl group to the acetyl carnitine. So acetyl CoA becomes CoH. So here we talk about this acetyl CoA. This acetyl is transferred to here, this carnitine. It makes acetyl carnitine by this enzyme, and this CoA is converted into CoH. Then this acetyl carnitines will go through these membranes and into the cytosol and and become a carrier for another long chain fatty acids. So this is all about the carnitine shuttle system and how carnitine is biosynthesized in our mitochondrial matrix. So I hope you people will understand. So this is all about my presentation. Thank you so much.